have Professor Mohammed, our guest speaker today. So welcome everyone to the biology seminar. So today, again, Dr. Mohammed Uddin is an associate professor at Mohammed bin Rashid University in Abu Dhabi, commonly known as MBRU. So Mohammed joined MBRU in 2017 after his academic training in Canada. Many years since doing his undergrads, then masters, then PhD, and postdoc. So he did his postdoc at SickKids at the University of Toronto with Stephen Scherer, who's one of the world experts in disease-associated CNVs and autism and de novo mutations. And I should mention that he did the last 2 years of his postdoc under a prestigious Bunting Fellowship in Canada. So Mohammed extensively published on autism and genetics, and would like to mention in particular a flagship Nature Genetics paper that he published in 2014. It was one of the really nice demonstrations of the role of de novo mutations in exons that are expressed in the brain. And for his work on autism in general, and also for several significant contributions to the field, Mohammed received the Nature Research Award for Excellence in Brain Research last year. And congratulations, Mohammed.
Thank you.
So more recently, Mohammed is extending his research to the use of machine learning and machine intelligence in neurodevelopmental disorders in general. And he's also very active in the COVID-19 research. It seems that everyone is doing something these days on COVID, and it's actually fascinating. The link between cognitive traits and COVID in itself is very interesting. So with that, welcome, Mohammed, and the screen is yours.
Thank you so much. You can hear me, right?
Yes.
Thank you, Youssef, for this nice introduction. And I really am very happy to be able to present at NYU today about my work. Before I start, I have to make a correction. I just changed the title from neurological to neurodevelopmental disorders, to be particular about the presentation. So this is my disclosure slide. So today, I'm going to first introduce very briefly the phenotypic and genetic heterogeneity of neurodevelopmental disorders. And then we will dive into the cellular heterogeneity of neurodevelopmental disorders that we see at the level of DNA and also at the level of cell transcriptome. And first, I will also introduce a very interesting story that came out from our research, where a germline mutation have been investigated at the research, and now it's been used for diagnostic in the clinic. So that's a very interesting story, at least to me, because of my involvement. And then we will talk about the postzygotic mutations that we see in neurodevelopmental disorders that was never been reported before. And then that's the genomic side of single cell omics. Then we'll talk about the single cell transcriptome, how this is becoming so relevant into the diagnostics and therapeutics of neurodevelopmental disorders. So just to briefly introduce the neurodevelopmental disorder, is a collection of rare disorders, and based on the rarity, we can see that the genetics of these disorders are also being rare. And if you look at the diversity of the disorders, you can categorize in two major domains that can be separated by functional variation. So the autism, epilepsy, ADHD, schizophrenia, all these disorders have functional deficits that can be observed at the level of synapses, whereas we have deficits like microcephaly, focal cortical dysplasia, double cortex syndrome. All these deficits can be observed in physical term. So there will be major brain tissue abnormalities because of these conditions. But all of these produce neurodevelopmental disorders of some sort. And the phenotypic heterogeneity starts from cognitive behavioral issues to severe seizure, and to SUDEP, which is sudden death. So it's a wide range of severity in phenotype. So throughout my talk, I'll basically point out two of the diseases from the neurodevelopmental disorder umbrella. So basically, I'll show you results from autism and epilepsy. And from twin studies, most of these neurodevelopmental disorders are shown to be highly genetic. So for example, if you look at obesity and diabetes, the genetic risk factor is fairly small, but where twin studies shows autism, epilepsy, ID have the heritability greater than, in some cohort, it is greater than 95, or in some cohort, it is 75. So the range is between 75 to 95, depending on what population you are measuring the heritability. So it's a strong genetic contribution to these diseases, and a wide spectrum of variation from karyotype, large, chromosomal level abnormalities like, trisomy 21 for Down syndrome, or CNVs that are 100 base pair or more in size that disrupts multiple genes, or at the level of single base pair variation. These are the wide spectrum of mutation that we see get mutated in neurodevelopmental disorder children. So these are mostly pediatric cases that we see in the clinic. The first two we can detect by clinical microarray, high resolution microarray, but the ultimate goal is to go for whole genome sequencing, so we can catalog the full spectrum of mutation just by doing one test. Currently, whole exome is in the middle of clinical microarray and whole genome sequencing. That also have a very good yield of diagnosis. So we are slowly transitioning to a whole genome sequencing. So I will start with the contribution of germline de novo mutation into neurodevelopmental disorders. So what do I mean by de novo is these mutations arise in the spermatocyte or oocyte of the parents. So they don't have this mutation in their body or in the other tissue of the body, apart from the germinal cells. And these mutations, when the zygote form, these mutation, the zygote carries on the mutation, so that mutation impacts all the tissue in the offspring. And it can be again SNV, indel, or copy number variation. So this, as I said, this is neurodevelopmental is a very big range of phenotype, and the spectrum varies on what mutation you're looking at. So this is one particular example of a syndrome, and we call it 15q13.3 microdeletion syndrome. That actually impacts about 0.4% of the global NDD population. I know it sounds small, but in neurodevelopmental disorders, it's a collection of few thousand disorders. So 0.4 is making it one of the prominent disorders. We have hundreds and hundreds of cases who have this microdeletion, and they manifest this wide spectrum of clinical symptoms that includes 28% of the kids have epilepsy or seizures. 16% have some sort of speech delay. Autism can be diagnosed in about 11%. Also we observed in schizophrenia, and intellectual disability is the most common denominator. About 57% of the kids have this manifestation when they have these disorders. But the main problem is this, 1.5 megabase length deletion have 10 genes within the boundary. And we have no idea what gene is the disease modulatory, gene. So if we have no idea about the targets. So if we do not know the targets, therapeutic or drug design based on the mutation becomes very challenging. Even for this deletion, CRISPR might not be a viable technology yet, because it's hard to, induce these large breakpoints and revert back the wild type is not been demonstrated yet. So we need to know the target gene to identify a marker for this disease. And again, this is the question we want to answer: which one of the brain causal gene? And to do that, we have administered an algorithm that we call critical exon gene algorithm. So what we have done, we took all the gene in the genome, and we identified the common breakpoints for the exons, and we have applied the 524 brain tissue transcriptome data from this spatio temporal data, and we took this data and mapped the RNase expression at the level of exon. And we also took the burden of mutation data from 60,000 exomes. This is taken from NOMAD. The database used to be called Exact. Now it's known as NOMAD. So they had, at that time, they had 60,000 exomes. So each of these, exon have a burden of mutation, non-synonymous mutation, and also we have observed the exon level gene expression. What we have found, this is a fundamental thing we have identified regardless of a disease. There are certain exon that accumulates a low number of pathogenic or, not pathogenic, I would say, protein disrupted mutation. But these are highly expressed in the brain. So it seems like these, exons are protected from accumulating mutations. And we observe this property is not been seen in all genes, but there are certain genes in the genome that have these exons, and they're highly enriched with these exons. And our initial observations shows that these pathogenic genes, SCN2A, Fragile X syndrome, FMR1, FMR2, SHANK1, SHANK2, these are all genes that are known for autism or other neurodevelopmental disorders. These genes have been identified to harbor a lot of these critical exons. So that means the function of this exon, functional unit, needs to be preserved for a neurotypical behavior. So this leads to the detection of the correlation that we see between these exons and the de novo mutation. So our question is, do we see de novo mutation lands in these exons, or the exons that are disrupted by mutation in normal controls and not much expressed in the brain? So what we observed, these exons are highly accumulating de novo mutation that we observe in autism spectrum disorders and also in other NDD genes. So what we took those 10 genes and we have mapped this critical exon, and what we have identified that this OTUD7A, an unknown gene, have a high number of critical exon, and it has multiple critical exon within its protein domains, and there is no accumulation, virtually no accumulation of non-synonymous mutation, but extremely expressed. And we have this spatiotemporal data, and we saw this gene actually is highly expressed in prenatal brain compared to early childhood and adult. And also this gene is extremely expressed in the prefrontal cortex compared to the visual cortex of the brain. So this kind of shows some correlation of the known biology of neurodevelopmental disorders, because the prefrontal cortex is very important in cognitive decision making, and also these diseases are manifest prenatally. So having a developmental gene make more sense for this microdeletion syndrome. But this doesn't necessarily directly prove that this gene is the causal gene for this microdeletion syndrome. So what we have done, we have conducted many other experiments. So we did this qPCR experiment in other tissue just to see if this gene is restrictively expressed in the brain or other tissue. It shows the brain, all these brain tissues that we have in our lab shows high expression of the gene compared to other tissues. So this gene is selectively expressed in brain. We also did WGCNA analysis with the human proteomic data. I'm skipping all this analysis because I think there's not much time for this presentation to present the methods. But what we have identified from the proteomic WGCNA that the OTUD7A gene is there's a first degree relationship with so many de novo mutation um, uh, protein. For example, the Fragile X targets also correlated with this gene, and also we have a database of autism de novo mutated genes, and their protein shows first degree relationship with this particular gene. So it adds, more value, more evidence to the story that this gene might be important for cognition, because it falls within this pathway. So what we did, we introduced CRISPR, and we have introduced a 9 base pair indel. 9 base pair indel because we had a patient who had a 9 base pair indel, but it was not diagnosed based on that exonic indel because this gene was not known. So the diagnosis was not done. But we followed our instinct based on our data from the in silico analysis, and we did the incision, and we have grown colony and converted into neurons at the end. And what we have identified after Golgi stain, and we have done extensive skull analysis, that this is the control and this is the wild type OTUD, and this is the deleted OTUD. So what you can see that there is dendritic spine of the neurons of our cortical neuron. It's highly reduced after the deletion. So this gene have a direct impact on the dendritic outgrowth. So if the dendritic outgrowth is reduced, that have many other implications. That means there will be less number of synapses within the brain. There will be no neurotransmitter going back and forth because of this lack of dendrites. And just to move forward with the study, so we have purified the wild type protein of the OTUD7A, and the overexpression also showed the rescue of this behavior. So sorry, when we overexpress the gene, we saw that the behavior has been rescued at the cellular level. So this kind of shows that a direct causal relationship of this gene getting knocked out and the lack of outgrowth of the dendritic spine. And just because that correlation, we thought that this might be linked with some postsynaptic area because of its relationship with the dendrites. So we have, and also this gene is not known to have a particular function. These ubiquitinase have a impact on the protein, but we have no idea where it's located. So we kind of located at the postsynaptic density region. So the protein is located at the postsynaptic neurons. So it kind of try to receive from presynaptic to postsynaptic to carry on function, but this outgrowth, lack of outgrowth will definitely show a detrimental effect. The interesting bit about this study, when we have these results, then we learn this other lab in Baylor. They have done a very interesting study, not like ours. I think ours is more interesting than this, but what they have done, they took these 10 genes and they knocked out most of the genes in different mouse strains. And they found that the mouse with this knockout shows the most interesting phenotype. And that's a validation for us. So both papers got published in the same journal, the same issue, even though we had no idea that this was been done in Baylor as well. We get to know in a conference when we're presenting and they're also presenting. So then we realized then we can co-submit together at the same time. So the journal accepted the paper, in the same issue. And the interesting thing is, this is one of the first paper that came out this year in January that shows homozygous variant within this gene that kind of like very confirmatory study for us showing that this gene is a causal gene for neurodevelopmental disorders. And they have shown that this gene is responsible for epilepsy and seizure. So now this gene is having a mutation, will get a diagnosis. Patient has now getting diagnosis based on the mutation on this gene. And this is not our group. I don't know this group at all. So, but they refer to our paper for the diagnosis. Is amazing. And this particular patient doesn't have any other mutation other than this. And homozygous mutation is, it's never been seen. And they also provided functional data supporting our evidence. So this is a very interesting story for me to show from research side how it can go to the bedside at the clinic, where what you discover can be, can improve diagnostics, therapeutics. So we kind of have this algorithm that can prioritize genes for downstream functional study. We have filed a patent and also licensed to a company who use this to prioritize genes, but we cannot stop there. So I asked myself, so what other mutation can be there other than this germline mutation that we see in neurodevelopmental disorders? So at that time, 3-4 years ago, that's the only type of mutation that's been associated with NDD. So what I was interested in, the idea came from some of the anecdotes that I was reading from a few cases. So I was interested in like cancer-like mutation, somatic mutation that produce this clonal complexity, how they are, impacting neurodevelopmental disorders. So as I said, the de novo mutation impacts the spermatocyte or oocyte, but the somatic mutation, after the zygote is formed, there are many stem cell or progenitor cells that are responsible to grow in different trajectory, germinal trajectory, and have the role in development of multiple tissues. So this particular progenitor cell might accumulate a mutation due to replication error or DNA mismatch error. And that particular stem cell and its progeny might be responsible for brain, and brain will accumulate that mutation. No other tissue will have it. So it's a very localized mutation that creates the complexity. So I wanted to investigate how this can be, translated into diagnosis, because it's not known to be impacting neurodevelopmental disorder or autism. So that's the question I wanted to answer. Can we detect pathogenic somatic mutation in NDD? So this is a case that we published, and I'm very excited to show you the results. So this is a kid who had severe epilepsy. He had seizure every day, 3-4 times, and he used to wear a helmet to save his head. He's a 6-year-old kid and sick kids, and MRI shows, even though it might not be clear from this image, there are flare signals coming out from a localized region in the brain. So the neurosurgeon took off the skull and installed a grid to identify localize the flare signal in sick kids. And the neurosurgeon then intervened and resected the brain tissue where the flare signals were coming. So what we did, I took the brain tissue and we stained it initially. And sorry, before I go to this, we actually did a targeted sequencing and found out that this kid have a de novo heterozygous mutation in STXBP gene. That is a bona fide epilepsy gene. So it's a heterozygous mutation, deletion, that's impacting and causing this disease. There is no question about it because it's been functionally proven. But the postzygotic mutation story comes now. So when we looked at the tissue, we saw the normal distribution of neurons are even, if you look at the rightmost corner image. But this is the tissue where we saw so many neurons never migrated to its neurocortical layers. It's clumped together. That's where the flare signals, the abnormal electric pulses are coming. And what we did, we genotyped the deletion into this particular uh, tissue and also in the patient blood and also the control tissue. And you can see the deep temporal white matter tissue have below 1 copy of the mutation. That means some of the cells have homozygous deletion with unknown breakpoint. So it has a mosaic structure of this complex deletion that kind of answers the non-migration of the cell. So they have totally different breakpoints of the deletion compared to the other cell within the brain. So this also led to more investigation, and what we identified that we looked at families who have multiple kids impacted by the same deletion or duplication or a mutation. So that kind of shows the evidence for germline mosaicism, right? Because the same de novo mutation getting transmitted in multiple kids, that kind of points to the direction that the germline mutation, either mom or dad have a germline mutation in the sperm or oocyte that's getting transmitted over and over again. And we also identified that this particular kid have a PARP1 de novo stop gain mutation. So this SCN2A also a very well-known gene, but for autism, but we see these two sisters have these phenotypic differences as well in the severity. So our investigation further led to the detection that the father have a mosaic SCN2A deletion in his blood. And also PARP1 also found to be 20% of the blood cell of the father have these deletions. So it's mosaic in the germinal cell as well. And we also found out that this kid also have a really damaging de novo missense mutation, 60% of the blood cells. So we have identified using ddPCR back in a few years ago. So it shows the clonal complexity between the cases and the parents, and how this can be, how this clonal complexity can contribute to the difference in severity. But this shows that the postzygotic mutation have a role in autism spectrum disorder. And we wanted to, from this kind of anecdotal examples, we wanted to look into the bigger picture. So we looked at 5,000, about 6,000 trios, and we identified a differential, read that accounts for a second allele, second alternative allele, because that points to that some cell have a second allele, so that they're a mosaic mutation. And we identified that all the de novo mutation that we have reported, about 5,000 of these, 7.5% of these de novo mutations are mosaic in nature. Okay, and these mosaics are, I would call them high-level mosaics. So it's about 15 to 20%, because we didn't have the read depth to go below 5 to 6%, because you need then high read depth data to detect that much level of mosaicism. So anyway, so this kind of shows that some of the pathogenic genes like SCN2A and other genes also be picked up from the study to harbor mosaic mutation. And this shows that not just germline, also postzygotic mutation contributes to autism and NDD in general. So there are multiple papers came out after this. And we published all these papers in multiple journals reporting our findings. And we are continuing to that line to identify clonal complexity in neurodevelopmental disorders. So that leads to my last discussion about transcriptomics and cell types. That's what actually I'm doing now. So from now on, there will be a lot of unpublished data. So this particular graph shows that we have a primary brain cell that was flow sorted with known marker. So we know that this particular cell is neuron, glia, or astrocytes. Then we looked at the STXBP gene, how it is expressed in different cell types, known cell types. And we looked at that STXBP is only expressed in neurons. And that is very exciting, because if this gene is only expressed in one cell type, that means having a mutation will not disrupt the other cells. It's practically not expressed at all for other cell types. And also that it has some implication for future drug design, because it needs to be targeted, because you do not need to bother the other cells where the protein is not present. So it's very restrictive in expression. So that's why we wanted to identify, is it just neuron that all autism genes are getting impacted? We want to identify all the cell types that been detected, that we can detect from the mutation that we have seen in autism spectrum disorders. So we have applied 30,000 single cell transcriptome from 3 distinct brain regions, and the data was obtained from Allen Brain Institute. And we have also curated from multiple consortium from our collaborators about 1,000 genes that have de novo loss of function mutation data. So that data came from about 40,000 whole exome or whole genome sequencing. Is not seen here, tabled here, but we eventually will map that into this single cell data. The single cell transcriptome data. So we have 3 distinct brain region: anterior, the cingulate cortex, the primary visual cortex at the back, and also we have the middle temporal gyrus. So these 3 regions. The single cell has been processed after obtaining from Allen Brain Institute. We used CRUT, a known software, to identify the clusters. So these are the clusters actually. And we also identify the differentially expressed gene by applying 4 very strict criteria that have to pass 4 different independent tests. So we applied multiple new techniques that I do not want to say in detail to identify constant clusters. So the clusters that are highly constrained do not behave differently, that needs to be preserved. So these shows that there are multiple of these clusters. Applying critical exon hour method, that shows there are multiple of these clusters are highly impacted with critical DEGs. And we also applied a PLI cluster-based, PLI-based method on the cluster. And interestingly, the same cluster shows also in PLI analysis. So we have multiple analyses supporting that these clusters are highly constrained for the brain. Then what we did, we mapped the de novo pathogenic losses, loss of function genes into these clusters, and we identified that multiple clusters, especially these are the top 3 clusters that are colored in different color for 3 different brain regions that are highly enriched for LOF mutation. But the missions are, there are other clusters that are also highly enriched, but the LOF mutation are highly enriched for these 3 clusters, which you do not know what they belong to, which cell type they belong to. And we also identified these clusters that are highly enriched for these mutations, applying those 500 brain transcriptome data, spatiotemporal data. Those clusters that are enriched for de novo mutation are highly active in prenatal brain and also in the prefrontal cortex, you can see here. So this is the message from this analysis, that the cluster that we see enriched for de novo mutation in autism also enriched in transcriptional activity in the prenatal brain compared to adult or early childhood brain. So we also looked at briefly how those clusters that are enriched for de novo mutation, what are the DEG gene stands for, in what pathway. It shows that synaptic pathways, there are also pathway involving cell growth and development, also metabolic processes. So there are different pathways that relates to brain function or brain development that are highly active in those clusters, cell type cluster. And then we did an extensive analysis to identify the cell identity. So because you have the cluster, but you have no idea what they belong to. And interestingly enough, these are the highly enriched cluster. All of this cluster, this is our major identification from this analysis, these are non-neuronal in nature. These are oligodendrocytes microglia astrocytes. There are also pathogenic genes known for autism enriched for these neuronal clusters. But these are very clean-cut association. We see these top 3 are all non-neuronal in nature. And we want to observe if this non-neuronal it stands or not with other data. So we have provided validation data, but we also want to see the known gene where they expressed and how they expressed in this particular cell cluster that we have. So these are SCN2A, as I showed you, CHD8, the gene that we know causal for autism. For example, SCN2A not expressed at all for this non-neuronal identity. So our identity really makes sense because SCN2A is a it's a neuronal marker. It produces a protein at the synaptic junction, and so it's only expressed in the neuronal cell type. CHD also is expressed in neuronal, but it has some non-neuronal expression. So what it shows that the known genes are some of these genes are strictly neuronal, or some genes are widespread because CHD is a chromatin remodeling gene. So it's very interesting results for us. We also then looked at those 3 brain brain cluster that are non-neuronal in nature. Is it true? So we kind of expressed the marker that are known to be non-neuronal in nature, and it shows that our cluster identity analysis was correct. These are known non-neuronal marker only expressed in non-neuronal clusters. So that are also enriched with top, those LOF mutation that we see in autism. So this is a specific example showing that the CANC1 mutation, this is a CANC1 gene. These are all de novo pathogenic mutation, and these genes strictly expressed in those non-neuronal genes, not in neurons. Similarly, Planck's B1 also strictly expressed in non-neuronal genes, not in neurons. So our analysis pipeline shows very interesting results, and it shows that new identification that autism just not happen due to neuronal bias, that there is a non-neuronal etiology into the gene. So we are currently preparing the manuscript, and this is some validation data. These are known cell type, expression for the CANC1. The cell types are known, and it shows again non-neuronal expression for the CANC1 neuron. This gene is not expressed, and interestingly enough, this gene, the expression pattern is preserved because we saw the similar thing that CANC1 is expressed in non-neuronal cells, not in neurons in mouse. So the fold change and the expression pattern compared to neuron is strikingly different in astrocytes for both of these genes. So it has an astrocyte angle to autism that's not been validated or shown before. So in summary, we have shown an algorithm to classify pathogenic mutations. We have shown that postzygotic mutations can impact cell lineage in NDD. Autism pathogenic mutation impacts both neuronal and non-neuronal cell types from our recent analysis. And some genes are very selectively expressed in, and some are widespread in different brain cell types. So lastly, these are 2 slides I want to touch base on the AI bit that we are working, because this is also unpublished work. So almost all the algorithms that we use to identify cell types, they use a predefined cluster number. These are supervised learning algorithm and produces a natural clustering that have no knowledge of any biological objectives. Then you reinforce your biological objective to the identified clusters. So, and each cell have this multidimensional transcriptome, right? The transcriptome can be seen at the angle from splicing, spatially how they are, time-wise, the velocity, how these single cells are, can be clustered in terms of velocity. So you can come up with those hypotheses, then you should be able to cluster. But TSNE and UMAP, whatever other algorithm you apply, They're very good algorithm clustering, but they are not taking into account into these objectives or hypotheses. So what we are trying to do here, we have developed a multi-objective, unsupervised, it's an adaptive genetic algorithm that is unsupervised in a sense that when we feed the data into the algorithm, it has no knowledge of how many clusters it needs to produce. But the only thing it accounts for is your objective. So we started with two objectives: how cells are can be clustered based on the fold change and the expression trajectory. So based on that, this is a result. I'm showing that known with known cell types, we have known cell types that were flow sorted, and it can identify the neurons, astrocytes, and all these. And then it also can identify those unknown cell types that we do not know how to interpret, but we have now this fold change and expression trajectory to interpret them. So that's a very interesting, I find, the interesting way to cluster. So we are finalizing our results in this segment as well. So it will be a very interesting tool working with the Genomark, a social platform, a company or biotech in Toronto. So we are working with them on this project. It's very interesting results so far. So I would like to also thank my postdocs, my collaborators in different places, and also the consortium that helped me doing this research that I have presented today. And thank you, Youssef and New York University, for inviting me to this presentation. So if you have any question, please let me know.
Right. Thank you, Mohammed. Andrea, can you maybe start my video? Thank you, Mohammed. I have many questions, but I'm going to keep it to two, and probably we'll follow up after with some discussions.
Yes.
So first question is about the interesting case of mosaicism that you showed, that case in the father. So I'm really intrigued by the fact that you see it both in, you see it also in blood, but it's also mosaicism. So that means that it's happening independently in different tissues, if I understood correctly.
Yeah, it's a very complex case, yeah.
So what's the mechanism for that? What can drive those, the same
so
de novo mutation independently in different tissues?
Yeah, so I think that's how I would interpret, even though I have no proof of it. The interpretation would be, brain follows the ectoderm lineage, right? So I think this mutation happened before the ectoderm originated. So before the ectoderm, this mutation also have a lineage for the blood and also for the lineage for the brain. And the lineage, that progenitor stem cell that have carried this mutation maybe not have branched out to other tissue. So you see this high level mutation in blood that might also impact the germinal tissue, maybe sperm of this father. And every time he mated, that just by chance, that particular sperm who have the mutation got transmitted. So this is a classic example, and we have actually looked at other family who have multiple kids with this type of mutation. And we have found that those mutations, a big chunk of those mutations are actually mosaic in father's or mother's blood. So is germinal gonadal mosaicism going on, and also you see it in other tissue. It's very complex scenario, to be honest.
I see. And is it often paternal origin, or
it is now? We think it is because sperm with age accumulates a bit more de novo mutation that's been shown in few studies. But there are also some contradictory studies there as well. But the de novo mutation, this has been validated multiple times, that is actually the origin is always 60 to 70% is paternal compared to maternal.
I see. My second question is about the very intriguing enrichment that you see of autism mutations in the non-neural lineages. So that's really intriguing, but that's also, could it be driven by some sort of mechanisms of
for example, yeah, I think this, we are actually about to submit the draft. It's better that you can, question me more on this. So what we see is neuron have the focus because of its property, how fascinating neuron is, and all the funding actually went towards neuron. But all these other primary cell in the brain left out to identify their role into different diseases. And if you look at astrocytes specifically, it actually helps form the synapses. So the synaptic formation not just happen due to neurons, it's also astrocytes are also involved in it. So having a mutation in a gene that impacts the astrocytes will also impact the synapse, which is a causal deficit for autism. And this is what we are observing. That's been, rigorously not been studied and not been reported. So I think this is one mechanism I can tell you from our analysis. There might be other mechanism that involves, because for example, these genes are also have some immune feedback, these non-neuronal genes. Several glia type genes are also related to inflammation. And inflammation is also very big symptoms in neurodevelopmental disorders. Definitely, there are pathways that we still haven't identified involving non-neuronal.
So although you have 30,000 cells, it's still n equals 1. Could this be something unique to this individual?
Oh, sorry, this is not n equals 1. I forgot to add the donors. The number of donors is about, I think it's 16. Yeah, it's multiple donors actually.
So and these are the mutations are shared among all of them, or
no, I'm talking about the single cell. The single cell data from multiple donors, and the mutations are that we the curated and mapped into these clusters, it's not shown here, are taken from about 40,000 exomes and genomes of autism spectrum kids. And this particular mutation that we shown as an example, these mutations are from independent autism cases, each of these mutations. These are de novo loss of function mutation.
I see. And so my last question is, so could this explain potentially penetrance, like whenever it's taken place, because you might have other more penetrant causal mutations in the other neural lineages?
Absolutely. This, for example, if you have this CANC1 loss of function mutation, that person can also have a double hit. A double hit model is a very known pathway for neurodevelopmental disorders, and that other model can also impact the neurons. So you have this multi kind of multi-faceted impact where neurons and non-neurons both are impacted. That also can happen for sure. Yeah, we have observed many of the cases that have just because the nature how DNA breaks, it can break into multiple places. We have observed multiple deletions that are pathogenic, and they have totally different genes and have different pathways, and we just don't know what cell types they belong to. So that's the study I'm interested in, especially finding out cell type for brain cell type for NDDs.
Okay. So any questions from the audience? Alex?
Someone's got their hand up, so I'm just going to allow King to talk. Hello, King?
Yes. Hello.
Hi.
Hi, Mohammed. It's a very interesting thing. I just want to confirm with you about the finding on, as also interested by Youssef, was about the non-neuronal cell transcriptome in the autism populations. The astral cells and oligo cells are derived in the late stage of neurogenesis and also further. So they have the same root. They are all have the same root with the neuronal populations. If you find, if you find these populations have unique mutations than the neuronal population, then that suggests these cells are really postzygotic and probably happen during mitosis or post-mitosis. What do you think? I know some of the epigenetic modification may happen specifically to this non-neuronal population, but most likely it may happen during the mitosis. What do you think this is likely to be happen? And the second, following this notice is, you found this, since it's a single cell mutation detection, do you find this, if some like of the gene and PLX complex and B1 genes, are these also mosaic or mosaic, or they are all completely mutant?
Thank you, Quinn. I think I'll answer the last question first because it's easier. We haven't found these two genes. We haven't found any mosaic mutation on them. These are germline mutation. But what we are showing here, they're restrictive in their expression. They are not expressed in all cell types, all brain cell types. They're very restrictive, and they're only expressed in these non-neuronal cell types that we see. For your other question, that it's because that's true, is there all these cell types are coming to the same route with the neurogenesis. They, even when they migrate, follow through the subventricular and ventricular zones, but and during the formation of these cells or migration, anytime they can accumulate mutation, right? And this has been actually one of the papers that shows that even some brain activity can lead to double strand break in a single cell, and that will induce, again, it's like a constant bombardment going on in the brain. And if you look at the gestation stage, the only tissue that grow fast is the brain tissue. So almost the growth almost is like a tumor growth. It's fastest growth tissue in our body that has the huge number of cells, and it's prone to produce mutation due to the cell division, I think, and it can be due to replication error or due to mismatch repair. So this kind of mutation actually been observed. There is a science paper came out a few years ago, actually, a single cell, two papers actually, that showed each individual cell in neuron have their own private mutation. So the postzygotic mutation is very widespread, and when it happened, that kind of shows the clonality. You can trace the lineage, and then that might help. But again, it is. It would be very good if we, what you just said, if we find a mutation that only in oligodendrocytes or astrocytes, not in other neuronal branch. But it's hard to find that kind of mutation.
Well, thank you. That's very informative.
Any more questions? So I have one more, Mohammed.
Yes.
So the accumulation of mutations in the ones that you showed, the enrichment, it seems to be really like probably in the first or even second exon, if I remember your slide. So how is it escaping nonsense decay?
I actually have no specific answer to that. The accumulation of the loss of function mutation, you mean, right?
Yes.
Yeah, so I absolutely have no answer to that, how it is escaping. It is a very active field of research, and we have confirmed many of these, mRNA with LF mutation at the mRNA level. We sequence the cDNA. We still see that it's not decayed. It's in the cell. It's there. We just don't know how it's escaping it. Yeah, that's a very interesting question.
Right, with that, thank you so much, Mohammed. It's
thank you so much.
exactly 1 hour, so it's great.
Thank you so much.
And yeah, and I'll follow up with you with some other interesting insights and questions.
Yes, please do. Okay, thank you so much.
Thank you.
And thank you, everybody. Okay, bye.
Thanks a
Thank
lot.
you very